This is Amanda Welch welcoming you to this Bite-Size Bio web seminar, which today is sponsored by GE Healthcare. Super resolution microscopy enables scientists to see things that have not been seen before. But is it a tool for everyone or just a few experts? Today, Liana Ferrand will focus on structured illumination microscopy, which not only enables new discoveries for generalists and experts alike, but is also the most appropriate tool for live cell imaging at super resolution. Today's presentation is titled Super Resolution Microscopy, Improve Your Imaging, Don't Reinvent It, and is being presented by Liana Ferrand, Product Support Leader for the Genomics and Cellular Research from GE Healthcare. Liana has worked for GE Healthcare since 2007. She started out as an application specialist for the Delta Vision microscope. In 2009, GE Healthcare, which was previously known as Applied Precision, licensed and commercialized the Delta Vision OMX Super Resolution microscope. Liana supported the Delta Vision OMX as the lead application specialist from 2009 to 2013 and learned how to utilize super resolution microscopy to acquire scientific data along with the global scientific super resolution community. Liana is an expert in structured illumination microscopy and has successfully imaged a wide range of live and fixed biological samples. Currently, she leads the cell analysis product support team for GE Healthcare and is globally responsible for educating the GE Healthcare application specialists and the GE Healthcare Delta Vision OMX customer base on super resolution imaging. Now, as always, we will have a question and answer session after the presentation. So please type any questions that you have into the questions box, which appears on the right-hand side of your screen, and I'll put them to Liana at the end. So now, over to you, Liana, for the presentation.
Thank you, Amanda. Today we'll talk about structured illumination microscopy, which is one of the super resolution microscopy techniques, specifically how structured illumination microscopy can be utilized to improve your scientific discovery instead of forcing you to reinvent the experiments you're doing on the bench. Okay? To really understand what I'm talking about when I'm referring to these super resolution techniques, I like to start at the basics and ensure that everybody is comfortable or familiar with the terms that I will be using and the definitions of them that I am implying. So if we start off, there are three main functions of a light microscope. Actually, any microscope, but we'll be talking about the light microscope series. Magnification, which is the ability of the microscope or objective lens to increase the size of our object or our biological specimen on our cover slip. Resolution, which is our ability to distinguish one object from another. And contrast, which is our ability to define the boundaries of any particular object within our sample. Contrast is essential in helping us distinguish one object from another, since we need to know the edge of one and the start of another. Until very recently, all advances in light microscopy were techniques to enhance our contrast in our images. All of the confocal technologies, all of the deconvolution algorithms, they were all attempts to help us improve our contrast so that we could be more confident in determining what we were really seeing in our biological images. Maybe about a decade ago, we started having discoveries in a field that has now been come to know as super resolution microscopy, and those techniques were finally helping to break down or overcome the resolution limit of light microscopy. So we're going to talk about this a little bit more. So what is that resolution limit of a light microscope that I'm referring to? If we talk about a known sample, so for example, these two discrete objects, they could be fluorescent beads, they could be two independent GFP molecules in our sample, and the way that we experience those at the detector or in our image is through these Gaussian intensity curves. So we don't see the discrete object, we see the light coming from them, we see these Gaussian curves with nice sharp peaks if these are infinitely bright. And if these two objects are separated, well separated by some distance we'll call d, these peaks are very well separated, and obviously we can determine that they are two unique objects. If they move closer together in our sample, then their peaks, their intensity peaks, these Gaussian-shaped curves, move closer together as well. And if they move so close but still have a little bit of a separation, we might still be able to determine that they are two objects, or the likelihood of them being two objects is still distinguishable. If they move closer again, those two Gaussian intensity curves join and become a brighter, broader single curve, and we can no longer tell that those are two objects in our microscopy images. This distance d, which they must be apart, can also be represented in a formula. It is calculated by the wavelength of the excitation light you're using divided by 2 times the Na, which is the numerical aperture of our objective. We can't really change the wavelengths that we're using because we're working in the visible light spectrum. So we can go UV to far red, so we can stick with the most favorable ones for our resolution, which are the lower ones. But there is also a practical limit to how far we can push Na. So we arrive eventually at our diffraction limit. This is another term for that resolution limit. And our diffraction limit essentially says molecules closer than about 200 nanometers apart will appear to be the same structure. If you're wondering where I just pulled 200 nanometers apart from, if we take the red excitation 568, for example, and the best wide field or non-turf objective that we can get, oil immersion, 1.42 Na, we do the math, and that equals 200 nanometers is our D. Okay? So that's our resolution or our diffraction limit. So what is super resolution? Another way to visualize what that resolution meant to us is in a voxel. So a voxel is our 3D pixel on our detector, and until now, our voxel shape would be 200 nanometers in X, 200 nanometers in Y, by 600 nanometers in Z. And if we were looking for some relationship between some green stain tagged proteins and a red tagged protein, if they fell within this voxel, we would see yellow on the image, and we would settle for saying that they are co-localized within our resolution unit of our instrument. If we now compare this to 3D SIM, so 3D structured illumination, our voxel size gets cut in half. So now we can resolve 100 nanometers in X, 100 nanometers in Y, and 300 nanometers in Z. And what does that do for us? It gives us an 8-fold volume resolution improvement. So if my red protein is in one voxel and my green proteins are spread in 1 or 2, I can now see truly the relationship, or more truly the relationship, of where red and green are associated with each other. And I can answer further the question, are they co-localized? Are they right next to each other? Are they inside of each other? Any of those kinds of questions you might be asking. Okay. So how did they do this? How did they arrive at super resolution? And numerous groups and numerous techniques were developed by a variety of research groups. So I've just told you we can't change this diffraction limit because we can't change the wavelengths that we're working in, and there's the physical limit to how far we push Na. Okay, so why not? Why couldn't we do this? We're basically stuck with the laws of physics, right? So instead, these very intelligent people started using those laws of physics to enable our imaging below the diffraction limit. Some of the techniques also utilize chemistry, but structured illumination is definitely a usage of physics, so I'm going to stick with that one for today. If we now look at some of the other super resolution techniques and the diffraction limit that they arrive at now, or how far they go past, for comparison, confocal and wide field microscopes are about 250 nanometers laterally and 550 axially. Structured illumination is twice that resolution, so 120 nanometers laterally and 320 nanometers axially for the green wavelength, for example, or 488 excitation. Localization microscopy is another super resolution technique capable of somewhere down in the 30 to 50 nanometer range laterally. And STED, stimulated emission depletion, is also capable of something like 50 nanometers in XY, and some new advances are allowing for about 300 nanometer resolution in Z. Okay. A different way to visualize this is I really appreciate this graphic from the Journal of Cell Biology in 2010. This is still one of the best representations of the three-dimensional point spread function you can expect in the different techniques. So this over here is the comparison of our confocal and wide field resolution units. And then we have SIM, STED, and localization. This is a little bit outdated because it is representing 2D localization only. There are researchers utilizing 3D localization at this point, so there are some Z resolution improvements here in those specific techniques. Okay. So what is this localization technique that I'm referring to? Essentially, how localization works is that you collect a series of many images over time with only a few of your fluorescent molecules on at a time. Then you use a Gaussian model, like that intensity curve we were fitting to our bead, to determine where the center point of that molecule is, put it into a text file, and then plot all those points to create a map, which is like our image for our biological representation. This can get you 30 to 50 nanometer resolution. It is complementary to immuno-EM, so it can get very small structures resolved, but it does require very specific sample preparation. This is one of the techniques that really relies on chemistry, hence this disadvantage requiring of the time-sensitive buffers. And really, this is very limited in live cell capability because of the amount of light you have to utilize to get the molecules to blink over time, and the duration of the experiments that you have to subject them to that light. STED, in comparison, is using a little bit of chemistry and a little bit of physics, and it is the only technique that does not require some kind of mathematic fitting or reconstruction. It is a confocal technique, and it uses two different laser beams to excite the fluorophores in your sample. So it does a traditional laser spot, and then it hits that same spot with a donut-shaped beam. And the control of the hole in that donut is how we get very small areas of excited fluorophores so that we can resolve much finer structures. This, having to use both of these lasers for every image that we do, it's a lot of excitation. The donut beam is actually quenching the fluorescence where it's hitting, so it's leaving that very small point spread function. So there is, again, a lot of light put into this sample. Early on, it was a two-dimensional technique, but you could get 60 to 70 nanometer resolution. There are now advances that are allowing some Z resolution improvements. Traditionally, it was the confocal resolution, so 500 nanometers. Now you can get 300 if you're using some of the advanced techniques. It does require some very special probes that will actually do what you need it to do. Photo bleaching, because of the laser power and the type of scanning and excitation you have to utilize. But it is tunable, so you can adjust this if you want. And it is, like I said, the only direct visualization or scanning technique that doesn't require some math afterwards. Okay. Moving on to structured illumination, which is what I'm much more familiar with myself and what we'll talk about for the remainder of the morning or afternoon or evening, whoever's listening at what time. How structured illumination works is that we illuminate our biological sample with a structured excitation pattern. So instead of a nice flat wide field sheet of light, we are now using a striped pattern. We also measure the optical transform function. So the OTF, I'll call that, is the point spread function, which is our expected behavior of light through our system represented in frequency space. We need both of those known things and our biological image to use math to extract the higher resolution information, the higher frequency information from our images that we collected on our detector. Why do we do this? We get a twofold increase in resolution in X, Y, and Z. We also experience a significant increase in contrast through this mode. There are multiple types of SIM modes that you can utilize. It can also be used to correlate with EM or other super resolution techniques, or even wide field or confocal techniques. It is a wide field technique, so it requires the least amount of light of any of the super resolution techniques at this time. I have the same advantage and disadvantage listed here because sometimes twofold increase in resolution is not sufficient for your structures, but many times it is, and we'll talk more about when those times are. With the math and the reconstruction, this technique is prone to artifacts, but they are well characterized, but it is something that we have to account for when we're looking at SIM data. And it is limited in the Z depth into the sample because it is a wide field technique. So we can go somewhere between 30 and 50 microns in, but anything deeper than that, we start to lose the ability to excite our fluorophores. So what are some of these questions scientifically that SIM can help us answer with our twofold resolution improvement? Or another way to say that is, when do you need SIM? Okay. We already talked a little bit about this idea, co-localization, with my graphic, a 3D voxel. So if you are currently looking at two proteins or two structures and you are using co-localization to say that they are so close together that they fall within my high-resolution microscopy resolution unit, you probably have more information to be seeking, and SIM is probably the easiest way to try to answer that question further. Some other areas which have helped SIM tremendously are these structural and diffraction limited samples. So are you asking where a protein localizes within the centrosome or the synaptonemal complex or the kinetochore? All of these structures have about the same size scale. They're around the diffraction limit of our high resolution microscopes, so they're somewhere between 100 and 200 nanometers in size, diameter, separation, and they are so anything like that size is pretty much the poster child for structured illumination microscopy question answering. Further from that, you might have these structural questions that you need to also resolve temporally, right? If you are trying to see where two proteins are localizing with respect to each other throughout the cell cycle, throughout some dynamic process, watching something in microbiology, some bacterial cell dynamics, you need to be able to get your reconstructed frames quickly enough so you can watch these structures over time at the super resolution visualization. The other topic, it's a little bit harder to explain, but immuno-EM is not always an option for some scientific questions. So if you need 3D data, EM probably falls short, or it becomes extremely tedious for you to try and answer your questions. Further from that, sometimes this goes the other way, and EM is suggesting additional information is to be gained. It defines a structure very nicely, but you need to know where proteins are localizing within that structure. So that might help point you towards SIM being a good technique to try next. Okay. An example I already pointed out, centrosomes was a good example of a structural and diffraction limited biological thing. Biological structure. And if we image these in a wide field microscope, and we have three different components of the centrosome labeled in blue, green, and red, we merge them all in the wide field image, and we don't really know what's going on with respect to each other here. We can sort of guess. We have some blue outside, we have green, what's going on here. We can deconvolve that, improve our contrast. It helps us define a little bit better the boundaries of these structures, but when we move to SIM, we can start to see there's a true separation here, there's red inside of a green ring here. This looks like now we actually have some Z depth of information here, and if you separate them out, you can see the ring structure. You can see even the red has a ring structure, and the red ring is inside the green ring. Okay. An example of the temporal and diffraction limited, this is looking at the FIT-Z ring in some bacteria, and we are looking, I'm sorry, the arrow, I'll leave my mouse here, the arrow went away here. So we're watching this ring-like structure forming live in wide field microscopy. Up until this point, they have been unable to tell if this was a ring, if this was a helical spiral structure, if this was moving around, something moving around the solid ring. And now you can clearly see the ring forming here and the dynamics of the proteins moving around that ring structure. Okay. Some further data to show where SIM has worked in fixed samples from Lothar Schmelle and Jürgen Neumann back in, I believe this was 2008 or 2009 from Munich. This is some of the earliest SIM data, and this was looking at the nuclear pore complex and lamin around a nuclei. Okay, so wide field, this was where we were stuck. Lateral resolution, axial definitely falls apart quickly here. And then if we look at the same nuclei in nucleus in SIM, then we can start to see a lot more detail. We can see that the lamin is not this solid structure or sheet inside of our nuclear pore complex. We can see that the nuclear pore complex is fitting within our lamin, and we can see that all around the nucleus here as well in our Z resolution. A different view from a different group in Basel of nuclear pore complexes, we can truly appreciate the 3 dimensions of resolution improvement as this rotates around, and we can see the different nuclear pore labeling. Okay. Another example of that structure, diffraction limited structure, these are cilia from the Shaw lab in Harvard Medical Center. And they are looking to determine what these two structures, how they interact with each other. And in wide field imaging and deconvolution, it's the same. You kind of get this yellow, and you know that they're really close together, but what's going on? In the SIM, you can start to resolve truly the ring structures, and you can start to see there's still some yellow, but you can see red and green, and maybe they're twisted around each other. You can start to further advance your answers to this question. Some cytoskeleton, so actin and microtubules, what they would look like wide field and 3D SIM. And this is a thick culture that I'll use to show that this was 20 microns of a yeast mycelium culture. It was cleared, but this is rotating through the volume, so you can see the depth and how we resolve the yeast. Okay. So how do we do this? How does structured illumination actually work? We take, if we talk about a biological sample, and I'm going to use my zebras here as my biological sample because they have some stripes of their own, so it becomes very easy to show you what I'm going to try to show you. If I overlay my structured illumination pattern, so striped pattern on my zebras, you can already start to see, maybe if we focus here, that those stripes on the zebras start to change a little bit, at least to our eyes. If I move those stripes over the zebras and look at their stripes versus what's happening, I'm starting to see that interference, and so we'll call that interference, those are moire fringes. We're essentially doing this on purpose in our biology. We're going to step our stripes or translate our stripes across the biological image, structure, and we're going to also, we're also going to rotate that around to 3 different angles while we rotate. So you can see that the 2 different directions of movement create different moire fringes. So we're going to do this on purpose to our biology. We're going to know our striped pattern. We're going to know how we expect light to behave, and we're going to be able to extract further details from your biology. Okay. How do we create this striped interference pattern? For comparison, if we talk first about what wide field excitation light path looks like, very simplified. We take some kind of light source, a xenon bulb, a mercury bulb, a laser, a solid state illumination source. We put it through a fiber optic cable into an optical path, eventually to the back aperture of our objective. It comes out of our objective, nice solid sheet of light, focused at onto our cover slip, onto our sample. And we get excitation that's nice and even. If instead I take that same fiber optic cable light input and I put it through a diffraction grating, that diffraction grating splits that beam of light into infinite beams. It will go on and on. We only have represented the zero center order beam and plus and minus first and second orders. This would really go on to infinity in positive and negative directions. But these three beams, orders of the beams, are what we're going to focus on. It does go through numerous other optics, but to simplify here, we're going to put those three beams into the back aperture or back focal plane of my objective to allow them to come through the objective and meet right at the sample plane to interfere with each other and create a striped interference pattern on our sample. So now this puddle of beads looks like it has stripes in it. It doesn't have stripes. It has constructive interference, which is the bright part, and destructive interference, which is the dark part. Okay. So once again, side by side, if I didn't move my sim pattern at all, this is what I would get excited, and I would be stuck with stripes in my image. Okay. How we collect sim data, though, is we translate that striped pattern, like I said, so we call those phase steps. So that's when we physically step the stripes across the image, and we do that at 3 different rotations. So it's a little bit hard to see, but the stripes are this way, and then this direction, and again this direction. So they're 3 distinct rotational angles, and we do 5 phase steps or 5 translations at each position. So for each Z section, for 3D Sim, we're collecting 15 images to reconstruct that single slice. We also have other sim techniques, and other people have, there's other sim techniques being utilized. There's 3D sim like we're talking about, or we've been talking about to now. There is 2D sim, and there is now very more recently this turf sim capability, okay? So the difference between the 3 modes is essentially what we're doing with these 3 beams that we sent through our diffraction grating. For 2D sim, we simply block this zero order beam, and we allow the first and positive first and minus first order beams to interfere, just like our 3 beams did, at our sample plane. For turf sim, we now move those same 2 first order beams out to the turf angle of our objective, and we create that interference pattern only in the turf plane. If anyone listening is not familiar with turf, it is a term called total internal reflection fluorescence, and it only allows excitation within the first 200 nanometers of our cover slip. Okay? This is a different representation, so this is an XZ view of how the illumination pattern would look if we could visualize it through your sample. So you can see in 3D, we have the bright, dark stripes, but we also have that pattern in the Z dimension as well. This is how we get that that third direction of twofold resolution improvement, because we're actually sampling our resolution in the Z direction as well. For 2D sim, we're only going to get lateral resolution improvement because we're only interfering in the lateral direction. And in Z, it's going to look the same as we go through the depth of our sample. Obviously, somewhere farther up here is going to fade away. Like I said, 30 to 50 microns we can image in. And then the turf sim, we will only be getting our interference pattern at this very first few hundred nanometers of our sample. Okay. So does it really work in live samples? This is what sim has become more popular again for, because now we don't just want some fixed samples. We don't just want a single time point. We want to see what these really dynamic structural events are. Okay. So before I get to a yes or no question, I would like to visit something I say on a very regular basis, that all microscopy, super resolution, confocal, wide field, all microscopy requires compromise, right? So we have to understand what we're trying to ask. So if we're looking at a fixed cell image or a fixed cell sample and we're trying to image that, we have a certain photon budget, right? So let's talk about how we have a certain number of photons in the bank, and if we are trying to use super resolution, we need to put a significant amount of our photons into our spatial resolution improvement. we probably don't have to consider too much phototoxicity because we've killed our cells on purpose, right? We fixed them. We do need to worry about photobleaching, so we can't ignore this entirely, but we don't have to waste a lot of photons thinking about this, right? If we also, we don't have to go quickly because, like I said, it's dead, it's on our cover slip, it's not moving, right? So we can probably put the remainder of our photons into going through multiple dimensions. We can go thicker through Z. We can go more colors in the same sample, more channels. We can, nope, T was supposed to go away. Sorry, not through T, but we can focus these photons on going through Z and on adding more and more wavelengths up to 4 in a normal SIM instrument experiment. If we switch over instead to what we would need to do for live cell imaging, and we want to get this spatial resolution, we can't take our photons from here and redistribute them, but we really need to pay attention to our phototoxicity. We really need to go fast enough that we sample the data appropriately. We don't alias the event we're looking for by being too slow, or we don't miss it or take too long to collect a single frame, and it moves in the time of our frame, so it blurs out and we see an artifact. So where are we going to take that from? It's probably are we going to go as far through Z? Are we going to do as many colors? How much time can we actually afford to collect? Okay, so early on, we did some great 3D SIM imaging. This is a Drosophila nerve cell dissected out and put live on the microscope, and this is very early 3D SIM live cell data, courtesy of the Alon Davis group at Oxford University with Richard Parton. And you can see the movie playing through and zooming in now on the 3D SIM microtubule bundles. And you can see not only the individual microtubules, but you can see the dynamic interaction of them as they form bundles and separate. Okay. Stepping this up a notch, looking at some mitochondria in green and the ER marker in M cherry, we were able to do 2 color live cell imaging for over 200 time points, and there was very minimal photobleaching as you watch through, and there was also these dynamic movements as we can watch these mitochondria slipping through the ER, the matrix of the ER. What better to prove that a cell remains live through imaging? This is a mitotic cell going through division. We caught this and we imaged this for every 90 seconds for 13 minutes, and we got it going through division. This is an EV3 marker, and you can see the difference in the contrast as well as the resolution if you kind of focus on the astral areas of the wide field image versus the 3D SIM image. But not all biology will tolerate 3D SIM. These were pretty aggressive experiments, and so we have to consider the other techniques. Can 2D SIM or TURF SIM? What can we do? TURF SIM is very exciting because not only is it the true super resolution technique for TURF microscopy people, now that it can look at live events over time, but it's opening up the door for people who have been utilizing EM and localization to correlate, to now add a dynamic component to their scientific discovery. Were you looking at EM and then using localization because your structures were so tiny to actually try and define where your proteins are? But now you want to know what those proteins are doing. So we can start watching them in the TURF SIM mode. This is an M cherry LifeX in heliocells staining, and we were watching something that EM people refer to as the actin nest. Apparently they see this all over the place in EM. It's this ring structure, which we've zoomed in here. Play through one more time. In normal TURF imaging, people have watched, things touch down and walk around, but never had anyone been able, that I know of, for the researchers that we were working with, had actually watched it form and saw the ring. So this was exciting. This is a very short movie, every 5 seconds for 5 minutes, but we did move on to discover some additional live cell capabilities of the TURF SIM. This is, again, it's heliocells. This is a GFP tagged caveolin, which is a a particular protein of interest or structure of interest we did this imaging at AQLM in 2015, but Justin Taraska's group at the NIH is particularly interested in this. And what they're interested in is these really tight clusters. So I'm gonna zoom in on those, and I'm going to, before I play through the movie, they're trying to track individual caveolin particles. And if you look in the turf image, which they do very great turf imaging, or this is not a great image when we zoom in like this, but this is a very good turf image. But if you look at this section, that's this same line of particles here, okay? Up until now, this was what they were able to resolve, and they were trying to collaborate with extensive tracking mathematicians to try and model this and guess where things are going. And what TurfSim has enabled them to do now is simply utilize existing tracking software and plug this type of resolved data in and track their molecules over time. Obviously, there's more to their story, but that's one example of how TurfSim is also advancing the scientific questions being answered. Okay. Another example of this, a very dynamic example, this is M-cherry-tubulin and GFP clathrin. So I'm showing the whole field of view right now. We image this cell in TurfSim every 30 seconds for over 3 hours. And I'm playing through it because you can see in many areas the dynamics, and over 3 hours this cell was still dancing around and happy. And zooming in on that bottom right corner, so I'm going to zoom in on this section here. And play through this. So if you are familiar with clathrin, I'm specifically calling out that these are clathrin plaques. Clathrin pits resolution limit is much smaller or much finer than we can resolve with SIM. But clathrin pits form plaques, which are also ring structures, and so that's what you're observing forming here at the cover slip surface, are these pits joining together to form plaques, come down to the cover slip, and get absorbed back into the cell. So if you're going to try super resolution imaging, especially on live cell imaging, what do you really need in that instrument? What are the features of a great instrument? They should all have 3D SIM capabilities. That's what we all started with. And for the ability to get something out of your data, even if 3D SIM, your samples are intolerant of 3D SIM, it should have a 2D SIM mode. If you're also looking at these very dynamic surface structures, you should have the turf SIM mode. If you're looking at live cells, you definitely need some environmental control. You'll need acquisition speed, so you need to go as fast as possible so that these dynamic movements are captured. You should consider that there is wavelength dependency on resolution, so the system should help you optimize resolution per wavelength so you can get the best possible data. It would also be advantageous to have a wide field mode, so very fast in case your structure is going so fast that SIM is not going to keep up, or if you want to see a comparison, ring turf or a turf mode is also advantageous, mostly because it allows you to start localization microscopy as well when you do arrive at a scientific question that SIM is just not quite answering for you. So you could start correlating the techniques. If you put all these things in one instrument, in our instrument, this is the Delta Vision OMXSR, these are all options of the Delta Vision OMXSR, and you can see how that instrument is going to really help you start improving your science and not force you to reinvent it. The not reinvent it part is we don't require special fluorophores if you're looking at these structures in a confocal or wide field microscope already. Toss them on a SIM, and we should be able to get some data quickly. Okay. I'm going to leave you with a quote that really resonated with me from Eric Betzig, Nobel Prize winner in 2014, on a super resolution technology webinar hosted elsewhere in July of 2015. He said, "The more resolution you ask for, the more pain you can expect. You want to use the method that gives you the poorest resolution but still gets you the biological answer you are looking for." This sounds like I'm telling you maybe not to do super resolution. That's not what I intend, but try the techniques that are going to get you the answer but not make you have undue pain or change too many things. And SIM is very often a very quick and easy approach for advancing your scientific questions. And you don't have to believe me. The OMX customer base has published over 115 peer-reviewed papers in 2015 alone, which can be quickly searched and found. Okay. And on that, I will turn it over to questions.
Thanks, Liana. That was an excellent presentation. We have a few questions from the audience. If anyone else has a question, please feel free to post it in the questions box that appears on the right of your screen. So we have a question from Rekha. I'm sorry if I mispronounce your names. So they ask which SIM technique will be suitable for, or would be suitable for colocalization?
For colocalization, well, any of them really. If it's I always recommend trying 3D SIM if it's a fixed cell. The 3 dimensions, the optical sectioning of 3D SIM would be ideal to help you. And it's if you can't get 3D, then 2D SIM is usually an option on the same scope. So I would recommend trying the 3D SIM for colocalized structures.
Okay. And then Beyond asks, how do you calculate the optical transfer function for different platforms?
Good question. So the optical transform function is actually you measure the point spread function. So you can use a diffraction limited bead and collect the point spread function through the microscope. And most microscopes, if they're using experimental or measured point spread functions like this, they have a software feature which has the math to convert that point spread function to the optical transform function. Those are the same thing, the same bits of information, but the PSF or the point spread function is in real space where we live, XYZ, and the OTF is that information represented in frequency space, which is where we do a lot of this math, because the math in frequency space is simplified and we can go between real space and frequency space easily. It happens behind the scenes for us.
Cool. Okay, so Robert has a bunch of questions, and so the first one, I think that actually you all have an app that would help him with this. So he asks, what refractive index oil would you use with Prolonged Gold, which has a refractive index of 1.46?
Okay, so the 1.46, right? So Prolonged Gold is around glycerol. With the Delta Vision product line, we offer an oil kit, and we actually now have, as Amanda was referring to, we have online a free immersion oil calculator. So the mounting media, which is your Prolonged Gold, is only one of the factors that you should be using to determine your immersion oil RI. Room temperature, or 37 degrees, the actual wavelength that you're optimizing for, and so I would recommend using our oil calculator. It's it's downloadable. It's in the App Store on most mobile phones, and you can plug in your imaging parameters, and it will suggest a recommended refractive index to utilize for your experiments.
Okay. And I'm going to actually send out some information about the immersion oil calculator that you all offer to the audience. So you should be getting that, so you can get that soon, and you can click on it and read more about it. And also there's a link to where to download it. And then I think, let's see. So then he asks, how do you determine the refractive index of your sample if the mounting media is a hardening type?
Actually, I'm going to say for 3D SIM, you should avoid the hardening types. You go through all of this trouble to make these beautiful samples, and if you use a hardening type, they basically turn your cells or your structures back into pancakes. And so I recommend using one of the mounting medias that you seal and remain soft for the preservation of the actual volume, so you can get the true resolution improvement in the Z dimension. Prolonged Gold, for example, it is designed to harden, but if you mount and then you seal it with nail polish, it will not harden, and so because you remove its access to air. So that is a good early technique, to get started on that. The challenge with hardening medias is depending on the media, the environmental air humidity, the amount of time you've exposed it to air, it can differ from the center of your cover slip to the edge of the cover slip. So even if it's been curing for 2 days, depending on the volume you've used, you might have different refractive indices across your cover slip.
Huh, neat. It always cracks me up the things that we end up using in lab, like nail polish to cover the mounting media.
Yeah, and a lot of people steer away from the bright nail polishes because I'm sure that they do this to some things. In my experience, I haven't really witnessed this or had this problem. People think that the colors quench, but I actually prefer the most metallic nail polish you can find. So the bright color with the sparkly stuff in it. Actually, the chemical bond formed by that nail polish is very solid. If you ask any person in your life that paints their nails, when you try to take that off of your actual nails, it's impossible, versus clear nail polish, which you wash your hands or dishes once and that clear nail polish is flaking off. So I steer away from the clear nail polish for if you want to preserve your samples for a long time.
That's a good tip. I never thought about that, but yeah, if I put on the metallic nail polish, it stays on a lot longer.
Right?
And now we have a question from Richard, and so he asks, what are some of the most common artifacts?
So the most common artifact is, some people call it honeycomb. It looks like a very structured, geometric hexagon shape, and very often you'll see it in the noise. If your SIM data was collected poorly, it can be overlapping your structures as well. The good news is there's really only one cause of this honeycomb. Well, you need strong signal to noise, but the honeycomb comes to the forefront of the image if we have some type of asymmetry in our data collection. So the three major reasons we would have asymmetric scattering of light in our sample would be poor refractive index matching of our oil, photo bleaching of the fluorophore through the data collection, or if we maybe by mistake set our Z sectioning to start in focus on one side and go all the way out of focus on the other side. Those three things are almost always the cause of that honeycomb artifact. And if you can look at your raw data, it's pretty easy to pull those to pull out which one is which and check those and reset and take a new image stack.
Okay, and so I just wanted to let everybody know that it looks like we're going to run past the hour mark, but we do want to get your questions answered. So if you have time, please stick around and we'll answer them. If not, you should be able to get followed up by email. So anyhow, you all should feel free to keep asking questions, but I wanted to let those of you know that have to leave that we will follow up on them. So now we have a question from Leopoldo. And he asks, is there any environmental requirement for great SIM, such as humidity or temperature?
So I'm not sure if that's for the sample or for the microscope, so I'm going to answer both, right? So if it's for live cell SIM, yes, you want to have really great humidity localized control at your sample with the CO2 percentage that your cells require, and stable maintenance of your 37 degrees. For the actual instrument room environment, it is very important for it to be thermally stable. So temperature fluctuation can be pretty detrimental to your instrument alignment. Humidity can also interfere with the way some optical coatings last, but in general, a good microscope room is what you need for the SIM instruments.
Okay. And Julio asks, could you use super resolution with 2-photon confocal imaging?
Interesting. This is the second time I've had this question in about 2 weeks, and I'd never thought about it before. And at this time, I don't think so. I'd have to think about that further. I would imagine someone would have been trying that in their labs if it was possible. So at this time, I don't think so.
Okay, and then it looks like Leopoldo has a follow-up question, or has another question, asking about fast live imaging. If it's only possible for the turf sim, or if it's also possible for 3D sim, and in that case, how fast can the imaging be?
Great question. So for 3D sim, we have to collect at least 1 micron of our Z-stack, and our Z-sectioning is 125 nanometers apart. So for a single 3D sim frame, we have to collect at least 135 raw data frames, and we can do that in a second. Okay? So that's 1 color, 1 micron through Z, 1 millisecond exposure time, which is very usable on many samples that I've seen. And so if you need to go thicker, obviously it will take longer, but it is doable. You just might have to consider how many frames you're going to collect or how frequently you collect frames, but that's why I showed that those 3 examples of the Drosophila nerve cell was quite thick. The ER and mitochondria was 1.5 microns, so that's how we got fast enough with those 2 colors. But that mitotic cell was through 9 microns, so we can do it quickly if your biology, if your biological question can fit within a second or 2 time frame of data collection.
Okay, and then we have a question from Sarah asking about are there ways to minimize residual stripe artifacts in SIM, or at least ways to confirm whether or not an observed stripe pattern is biologically real or an artifact?
Residual stripes, you should call your service engineer, because your phase stepping or that translation of stripes is probably not happening accurately enough or big enough. So that's when we see residual stripes. If you're not sure if you're seeing something biological, like myosin has stripes in it, and you can see that, and if they're oriented properly, it can trick us into thinking we have residual stripes. If you go back to the raw data, what I like to do is, if you can, in a lot of software tools, you can actually average the 5 phase steps or the 5 translations of a single rotation at a single Z-plane. And if you can do that, in our software, we have a tool that does this for you, angle by angle. And if in your raw data, if you do this averaging out and you see stripes in that raw data, then it is, again, call your service engineer and have them tweak the alignment of your system, because you should not see that in your raw data. But that is the most sound practice if you think you might have biological stripes, to rule out or prove that they weren't there from the instrument, if you go back and look at the raw data and ensure that you didn't have them there, if you average out.
That makes sense. You want to rule out anything mechanical before you start looking into anything biological.
Mm-hmm. Yep.
And then Rekha asks, is there anything special? Is there a special step needed for sample preparation?
So, no. Well, good sample preparation is required. I've already, given my spiel about hardening medias. I don't recommend them. I would use the soft mounting media. SIM is really advantageous in that all of the common dyes and fluorescent proteins, as long as it matches the excitation lasers on your SIM system, they should work. If you're using the correct number 1.5 cover slip, those should work. The number 1.5 cover slips is an absolute must, but general good practice for sample prep is what SIM requires. And about a year ago, we hosted another webinar by Petrina Pellet, one of our field application scientists here on Bite Size Bio, called practical considerations for super resolution imaging. So if you want to know more about sample preparation, I recommend that webinar, which is from early May 2015, which maybe Amanda, you can send out, or?
Yes, that's what I'm looking up now.
Okay.
Okay, and then our next question is from Parminder, asking about what type of microfluidic system should be used for live cell microscopy.
That is tough. So the covered glass bottom Microfluidics chambers are what really needs to be utilized if you're doing high or super resolution microscopy. And the specific ones that your system will work with kind of depends on the stage that you're trying to mount them to. So some of the microfluidic chambers are like the 96 well plate format, so they're rather big. Those don't fit on the OMX stage, for example. Some of the slide-based ones, they would fit on the OMX. So if you need to do microfluidics, I would definitely look into the cover glass bottom chambers and look at your instrument that you're going to be utilizing and pick the one that fits your stage and works with your biology.
Okay, yeah, that works. And then James asks, what about the sim? So James is still around. Maybe he can clarify this because he's asking, what about the sim artifacts you mentioned at the beginning of the talk? So I think we'll let James clarify that, and then we'll move on to the next question, which is Parminder is following up and asking if EGFP is a good label for protein localization studies in bacteria, and then if not, which genetically encoded probes are the best?
So EGFP is a great one. I mean, it was one of the most used ones. There are some brighter ones. I believe M neon green is becoming a preference for people. But really, what I've heard advised, I was actually just teaching at the Analytical Quantitative Light Microscopy course. The best advice I heard was try a few with your cells and see which ones work and use the one that works the best. And by work, it's, it transfects the most cells, it's the brightest, it's the most stable in terms of photo bleaching. So EGFP is usually a really good starting point. I also recommend doing some quick searches of literature. Because of localization microscopy, there are a lot of review papers characterizing many different fluorescent proteins and fluorescent dyes and how they behave. So expected brightness, stability, performance. So if you want some other ideas, I would definitely refer you to a quick Google literature search.
Okay, and I think that is our last question. So we're going to go ahead and wrap up the end of the seminar. I want to thank everybody for sticking around and for asking such great questions. And thank you, Liana, for our very illuminating presentation and a great discussion. And thanks also to our sponsor, GE Healthcare. And finally, thanks to you, the audience, for taking the time to attend and listen in and ask such fantastic questions. If you've enjoyed the seminar and would like to view the video recording of the session, please visit the seminars page on bitesizebio.com. It should be available within the next 24 hours. There, you can also see the other webinars we have lined up for you in Bitesize Bio's Webinar Festival. So until next time, good luck in your research and goodbye from all of us at GE Healthcare and Bitesize Bio.